12.6.2 Biological nitrogen fixation. Very few living organisms can utilize the nitrogen in the form N2, available abundantly in the air. Only certain prokaryotic species are capable of fixing nitrogen. Reduction of nitrogen to ammonia by living organism is called biological nitrogen fixation. The enzyme nitrogenase, which is capable of nitrogen reduction, is present exclusively in prokaryotes. Such microbes are called N2 fixers. The nitrogen fixing microbes could be free living or symbiotic. Examples of free living nitrogen fixing aerobic microbes are Azotobacter and Bazarenica kia, while Rhodospirillum is anaerobic and free living. In addition, a number of cyanobacteria such as Anabena and Nostoc are also free living nitrogen fixers. Symbiotic biological nitrogen fixation. Several types of symbiotic biological nitrogen fixing association are known. The most prominent among them is the legume bacteria relationship. Species of rod-shaped rhizobium has such a relationship with the roots of several legumes such as alfalfa, sweet clover, sweet pea, lentils, garden pea, broad bean, clover beans, etc. The most common association on roots is as nodules. These nodules are small outgrowths on the roots. The microbe Frankia also produces nitrogen fixing nodules on the roots of non leguminous plants. Example Alnus. Both Rhizobium and Frankia are free living in soil, but as symbionts can fix atmospheric nitrogen. Uproot any one plant of a common pulse just before flowering. You will see near spherical outgrowths on the roots. These are nodules. If you cut through them, you will notice that the central portion is red or pink. What makes the nodules pink? This is due to the presence of leguminous hemoglobin or leg hemoglobin. Nodule formation. Nodule formation involves a sequence of multiple interactions between rhizobium and roots of the host plant. Principal stages in the nodule formation are summarized as follows. Rhizobia multiply and colonize the surrounding of roots and get attached to epidermal and root hair cells. The root hair curl and the bacteria invade the root hair. An infection thread is produced carrying the bacteria into the cortex of the root, where they initiate the nodule formation in the cortex of the root. Then the bacteria are released from the thread into the cell, which lead to the differentiation of specialized nitrogen fixing cells. The nodule thus formed establishes a direct vascular connection with the host for exchange of nutrients. These events are depicted in figure 12.4. The nodule contains all the necessary biochemical components such as the enzyme nitrogenase and lag hemoglobin. The enzyme nitrogenase is a molybdenum iron protein MOFE protein and catalyze the conversion of atmospheric nitrogen to ammonia. The first stable product of nitrogen fixation. The reaction is as follows: N2 plus 8 electron plus 8 H plus 16 ATP arrow 2 NH3 plus H2 plus 16 ADP plus 16 P1. The enzyme nitrogenase is highly sensitive to the molecular oxygen. It requires anaerobic conditions. The nodules have adaptations that ensure that the enzyme is protected from oxygen. To protect these enzymes, The nodule contains an oxygen scavenger called leg hemoglobin. It is interesting to note that these microbes live as aerobes under free living conditions where nitrogenase is not operational. But during nitrogen fixing events, they become anaerobic, thus protecting the nitrogenase enzyme. You must have noticed in the above reaction that the ammonia synthesis by nitrogenase requires a very high input of energy, 8 ATP, for each NH3 produced. The energy required thus is obtained from the respiration of the host cells. Fate of ammonia. At physiological pH, the ammonia is protonated to form NH4 positive ammonium ion, while most of the plants can assimilate nitrate as well as ammonium ions. The latter is quite toxic to plants and hence cannot accumulate in them. Let us now see how the NH4 positive is used to synthesize amino acids in plants. There are two main ways in which they can take place. First one is reductive amination. In these processes, ammonia reacts with alpha ketoglutaric acid and forms glutamic acid as indicated in the equation given below. Alpha ketoglutaric acid plus NH4 positive plus NADPH arrow glutamate or dehydrogenase glutamate plus H2 plus NADP. Second one is transamination. It involves the transfer of amino group from one amino acid to the keto group of a keto acid. Glutamic acid is the main amino acid from which the transfer of NH2, the amino group, takes place and other amino acids are formed through transamination. The enzyme transaminase catalyzes all such reactions. For example, amino donor, amino acceptor. The two most important amides, asparagine, and glutamine, found in plants, are a structural part of proteins. They are formed from two amino acids, namely aspartic acid and glutamic acid, respectively by addition of another amino group to each. The hydroxyl part of the acid is replaced by another NH2 negative radical. Since amides contain more nitrogen than the amino acids, they are transported to other parts of the plant via xylem vessels. In addition, along the transpiration stream, the nodules of some plants, example soybean, export the fixed nitrogen as ureids. These compounds also have a particular high nitrogen to carbon ratio. Figure 12.4, soil particles, root hair, bacteria. Hook, bacteria. Infection thread containing bacteria. Mature nodule. Development of root nodules in soybean. A. Rhizobium bacteria contact a susceptible root hair, divide near it. Successful infection of the root hair causes it to curl. C. Infected thread carries the bacteria to the inner cortex. The bacteria get modified into rod shaped bacteroids and cause inner cortical and pericycle cells to divide. Divisions and growth of cortical and pericycle cells lead to nodule formation. D. A mature nodule is complete with vascular tissues continuous with those of root. Figure 12.5. Substrate nitrogen gas into enzyme nitrogenase, binding of substrate, reduction plus 2H, reduction plus 2H, reduction plus 2H, product ammonia NH3, release of products, free nitrogenase can bind another molecule of N2. Steps of conversion of atmospheric nitrogen to ammonia by nitrogenase enzyme complex found in nitrogen fixing bacteria.